My congratulations to Joanna Fowler and the chemistry laboratory at the Brookhaven National Laboratory for this honor being bestowed on them by the American Chemical Society. It is well deserved, and I regret that age and health issues prevent my direct participation in this celebration, but I hope that this videotape will be an acceptable substitute. 1944, Vannevar Bush, President Roosevelt's science advisor during World War II, sent him a memorandum which contained the following passage, which is shown in the slide. After the war's end, in 19 actively campaigned in support of the idea that a rich nation like the USA could and should, in its own best interest, support scientists to conduct basic research of their own choosing and curiosity. And this would ultimately prove beneficial to the country and to humanity. The many fortuitous circumstances in the development of the 18 FDG method and its applications are, I believe, an excellent example of the validity of his vision. As a participant in this history, I would like to briefly review the background and the history of its development. In 1948, Seymour Ketty and Carl Schmidt published their nitrous oxide method for the measurement of average rates of blood flow and oxygen consumption in the brain as a whole in human beings. Applications of this method demonstrated changes in the brain's energy metabolism in conditions in which consciousness was depressed. But it failed to detect any changes in conditions in which normal brain functions were certainly altered, such as normal sleep, performance of arithmetical calculations, and a number of other conditions. This was in contrast to findings in other organs in which altered functional activities were clearly associated with changes in oxygen consumption and blood flow. A popular explanation for the failure to find the same in brain was that the energy required for specific functions in the brain was negligible compared to the energy consumed by the baseline housekeeping functions of the brain. Ketty and his group, however, hypothesized that specific functions are localized to specific regions of the brain, and that when they are altered, their effects on metabolism and blood flow are localized only to those regions and diluted out in measurements of average rates of blood flow and energy metabolism in the brain as a whole. What was needed, we believe, was a method to measure energy metabolism and/or blood flow locally in the appropriate anatomical regions of the brain. Functional activity is localized to specific cells, for example, neurons, which do not have their own private blood flows. Blood flow supplies a wider area surrounding the cells, and furthermore is an essential and the almost exclusive substrate for the brain's energy metabolism. And its consumption is almost completely stoichiometric with the brain's oxygen consumption. I therefore made an attempt to use radioactive glucose and the quantitative autoradiographic technique to measure local rates of glucose utilization in the brain, but failed because the labeled products of glucose metabolism, which are carbon dioxide and water, were lost too rapidly from the brain. In 1957, I learned that high doses of the glucose analog, 2-deoxyglucose, caused a coma very much like insulin-induced hypoglycemic coma. Soles and Crane had previously reported that 2-deoxyglucose could be phosphorylated to 2-deoxyglucose 6-phosphate by glucose hexokinase, the enzyme that catalyzes the phosphorylation of glucose to glucose 6-phosphate, the first step in the glycolytic pathway of glucose metabolism. Both glucose and deoxyglucose cross the blood-brain barrier and are converted in the brain by hexokinase to their hexo-6-phosphate derivatives. Normally, the glucose 6-phosphate is isomerized by glucose 6-phosphate isomerase to fructose 6-phosphate, that radioactive, which is then metabolized down the tracer doses to low blood glycolysis and lactate, might be used with a quantitative autoradiographic technique to measure local rates of glucose phosphorylation and thus glucose utilization in the brain. Differences in enzyme kinetics between glucose and 2-deoxyglucose phosphorylation by hexokinase would of course have to be addressed. My lab was then fully engaged in studies of the biochemical mechanisms of action of thyroid hormones, and so I filed the idea away for future study. In 1964, Martin Rivich joined us at NIH, where he modified the I-131 trifluoroiodomethane autoradiographic local blood flow method for use with a non-volatile 14C labeled tracer. He used 14C antipyrine as the tracer, which required adapting the quantitative autoradiographic technique for use with carbon-14. The availability of quantitative carbon-14 autoradiography resurrected the idea of using a 14C labeled 2-deoxyglucose to measure local cerebral glucose utilization. Shortly after he returned to the University of Pennsylvania in 1966, Rivich and I initiated a collaboration to develop such a method. The initial experiments were done with brain slices in this lab, and they confirmed that the approach was feasible. By 1974, a model based on enzyme kinetics was designed from which an operational equation was derived, and a practical method using 14C autoradiography that was developed. The method was first applied to rats and monkeys. As with the autoradiographic blood flow methods, the optical densities for the various tissues in autoradiograms closely reflected the local rates of glucose utilization. The autoradiograms, therefore, provided visual representation of the local rates of local glucose utilization in the brain. Applications of the method demonstrated dramatic changes in glucose utilization in brain regions with altered local functional activities that were easily visualized in the autoradiograms. The program was used in collaborative studies of visual functions in the monkey carried out with Mort Mishkin's Laboratory of Neuropsychology at NIMH. It was first presented publicly at the annual meeting of the Society for Neuroscience in St. Louis in 1978 and was sensationally received. In fact, Chemical Engineering News, because it required autoradiography of brain slices, not possible with humans. After its successful use in rats and monkeys, Martin Rivich raised the question of adaptation. This instrument consisted of an array of scintillation counter detectors that scanned the human brain in vivo for gamma radiation and reproduced images of the local isotope concentrations in cross-sections of the brain. The oxyglucose, however, contains only hydrogen, oxygen, and carbon. Hydrogen has no gamma-emitting isotopes, and oxygen and carbon have gamma-emitting isotopes, but with half-lives that seem too short for organic synthesis of labeled deoxyglucose. Serendipitously, I belonged at that time to a wine tasting club of NIH biochemists. One of them, Peter Goldman, was working on enzyme actions on fluorinated analogs of natural substrates. And I knew of his finding that glutamic decarboxylase, the enzyme that decarboxylates glutamate to form the natural neurotransmitter gamma-aminobutyric acid, or GABA, could also convert fluoroglutamate to fluorogaba. The explanation was that because the fluorine atom is so small, when it replaces a hydrogen atom in a non-critical position in the substrate, the enzyme acts on it just like on the natural substrate. This suggested that we use a gamma-emitting fluorinated analog of 2-deoxyglucose. On searching the handbooks of radioisotopes in my office, we found 18F, a gamma ray emitter with a 110-minute half-life. Now the problem was how to synthesize fluorine-18 labeled fluorodeoxyglucose. Rivets returned to Penn with this idea, and he and Kuhl organized a meeting at Penn to discuss the possibility of using 18F labeled fluorodeoxyglucose with Kuhl's scanner. Al Wolf and Joanna Fowler, radiochemists from the Brookhaven National Laboratory, were invited and attended. A fluorine bonded to the one carbon would be unstable, and so we decided on the next most distant carbon, the 2 carbon. Wolf and Fowler were confident that they could synthesize 2,18F2-deoxyglucose D-glucose at 2,18FDG. We asked them, however, to first synthesize a 14C labeled 2-fluorodeoxyglucose so that we could examine it and find out if it was phosphorylated by brain hexokinase and if it behaved like 2-deoxyglucose in the rat brain. They did so, and we used it to confirm that 2-FDG was as good a tracer for glucose metabolism in the brain as 2-deoxyglucose. Wolf and Fowler succeeded in developing a practical synthesis of 2,18FDG, produced it in their lab in Brookhaven, and sent it to Penn, where Rivets and Kuhl successfully used it with Kuhl's Mark IV scanner to measure for the first time regional rates of glucose utilization in the human brain. Soon afterward, Kuhl moved to UCLA in Los Angeles and took Michael Phelps and Ed Hoffman with him. Both had been graduate students with Michael Terpagosian at Washington University, where they developed the first positron emission tomographic PET scanner, and they soon acquired a PET scanner at UCLA. Phelps, Hoffman, and Kuhl then proceeded to adapt the 18FDG method for use with PET scanning, and they successfully applied it in a variety of normal and pathological human conditions. The publication of many applications of the 18FDG method by the UCLA group and in now commercial availability of PET scanners led to a proliferation of centers that carried out many outstanding studies, including the Classics Chemistry Laboratory in Norbolca Laboratory, in which they used the 18FDG method to identify the sites and some of the mechanisms of drug addiction in the brain of addicted patients. Glucose is consumed more rapidly in tumor tissue than in normal tissue, and the more transformed and malignant the cells, the higher the rate of glucose utilization. Giovanni De Chiro at NIH was the first one to exploit this trait to localize and grade the malignancy of brain tumors in human patients by means of 18FDG and PET. This biochemical abnormality in tumor tissue is now the basis of the wide use of the 18-FDG method to diagnose, localize, and stage a variety of primary and metastatic malignant tumors. 18-FDG, I have been told, is now the most commonly used radiochemical in nuclear medicine and is of great value in the practice of oncology and clinical medicine. When the 2,14-C deoxyglucose method was first presented at the annual meeting of the American Society for Neurochemistry in 1944, the eminent neurochemist Norman Radin remarked about what a crazy idea it was to use an inhibitor of a metabolic pathway to measure the rate of flux through that pathway. It was indeed a very crazy idea. And yet, who could have foreseen that pursuing so basic a question of whether functional activity in brain uses energy would eventually lead to such practical benefits to the practice of medicine and to mankind. What a confirmation of Vannevar Bush's wisdom. Sorry that I can't be at this symposium, our conference. I would have loved to be there, and I hope that everything goes well, and my best congratulations to Joanna Fowler, Nora Volkow, and all the others at the Brookhaven National Laboratory. They well deserve this honor. Thank you.